Hello everyone, my name is Il Park, and this is a lecture on principles of neural coding. Okay, so in a neural system, there's a spatiotemporal pattern of neural activity that's evolving over time that's processing some information, right? And what we really want to know is what is that information? So the neural coding problem is trying to study that internal representation of information by relating it to different variables. And typically we study it in a short time scale in the order of seconds or so. Can be shorter, can be longer, but this excludes the study of long-term memory or learning that lasts for many, many days or years. So these study of neural code are typically done very empirically, right? You would measure certain neural signals. Perhaps you inserted some electrode array, or maybe you're measuring calcium concentration somehow, or some other method to observe these internal neural signals. Now you want to give them meaning. You want to know what they're representing. So we typically try to relate them to different variables. For example, it could be a sensory system. You're studying visual cortex, for example. You show some movie. Every time I show this movie, the spatiotemporal pattern of activity is typically, as such. That could be a neural coding study. Could be many different kinds of sensory signals, not just vision. It could also be a cognitive process or variable. It could be working memory task where you showed something and the animal has to remember that 5 seconds later. So during that 5 seconds, you know that some information about that task is somewhere in the brain. It could also be decision-making or perception that are cognitive variables. It could also be intention to move or planning motor behavior. That signal also is going to be related into future behavior. So if you have collected such data sets, typically the approach are either of two directions. You could either study the direction where you go from these red variables, the external experimental task variables or measured behaviors, to the neural representation. This is called the neural encoding studies. Or you can go the reverse direction, and based on the neural signals, you can try to read it out. You try to decode what the animal saw or what the animal will do. So in this lecture, we will study several principles of related to the studying of this neural coding problem. And just to ground ourselves to a particular modality of signals, we will be talking about electrophysiology. So most of the time, we're going to talk about these spikes. These are the events, the sequence of action potential a single neuron would emit. We will be detecting it through, let's say, an electrode array. And what you see here is a plot of these timing of action potentials. So each tick here, this vertical bar, represents a single action potential fired at that particular time point. So there are many, many spikes over time here. And sometimes I'll play this as a audio. So here's an example of the sound. You'll hear 2 types of beeps, which indicate this blue dot here, this beginning of the trial. And every second there will be a beep just to give you a tempo. And the crackling noise that you hear would be the spikes. Here we go. These are just identical spikes that are spike sequence that are repeated. Okay, so now that we know how to read these kind of plots, the first principle that I would like to introduce to you is the principle of neural variability. Neural signals are very variable. So suppose you have a single retinal ganglion cell. This is the output neuron of the retina that sends spikes to the brain to be interpreted in later stages. And this is a single neuron that you're recording from, and you showed a movie to the animal, well, to the retina in this case, and that you showed the exact same movie. So this is a frozen movie that was showed 30 times. And the movie was just some random noise. It doesn't matter what the actual content of the movie was, but it's precisely controlled experimentally to be exact repeats of each other. But as you can see from here, in this raster plot, for each trial. The first trial, you have a bunch of spikes. The second trial, you have a different set of spikes. There are differences across these trials, right? I will sonify this and play to you different trials in a sequence. Here we go. Okay, hopefully you could hear the differences between different trials. Although they are similar, they tend to fire more during the first part of the trial than the later part. Each trial has different firing patterns. So there's huge amount of changes from trial to trial, despite the fact that the stimulus is the same. So if you could do this kind of experiment where you're repeating identical trials, it could be sensory stimulus like what we just saw, or it could be behavior. It could be a stereotypical behavior. Maybe the monkey was reaching a particular point in space repeatedly, right? As close as possible to repeating exact behavior. And what you would see is that the response from trial to trial would vary. And this is often referred to as noise. I'm putting it in quotes because noise is in the eye of the beholder. If it's noise to you, it doesn't mean it's noise to somebody else or some other neuron. But oftentimes you would call this a noise because the signal itself has not changed. We give the exact same signal, but the response changed. So whatever that was not signal. So we call it noise. Whatever is not signal is noise in some context. So here are some examples of this trial-to-trial variability from a different brain area. This is the area called MT, the middle temporal area from a monkey. It's a small, tiny area here that really likes visual motion. If whenever something moves in their visual fields, these neurons go nuts. So the stimulus here is called the random dot display kinematogram, where each dot here is displayed, according to a certain movement. And these responses here, as you see, would be this neuron's firing pattern. Right, so you could hear, hopefully, a huge amount of difference across the trials. So what are the sources of this trial-to-trial variability? It's because it's not the signal that we are trying to control in these experiments. So there are several biophysical reasons why we might expect to see these kinds of variability. In the cell membrane, biophysically speaking, there are ion channels that are stochastically opening and closing because we're not at absolute zero temperature. These are molecular machines. So there's a high amount of noise, thermal fluctuations in these ion channels that causes membrane voltage fluctuations. There are a huge amount of noise in the synapse because even if an action potential reaches a synapse, it does not always release a vesicle, and it does not always release the same amount of neurotransmitter. Right? The probability that you will release a neurotransmitter or a vesicle will fuse with a membrane is actually pretty low. In unreliable synapses, it could be as low as 10% or lower. So huge amount of variability. So every time your signal comes in, it's transmitted through spikes. Somewhere in the chain, some of the synapses are failing to actually transmit signals across the membrane. And then, of course, there are more intracellular signal transaction pathways and amplification, and all of these biophysical mechanisms that are inherently variable. That has nothing to do with high-level variability in the system. It's very, very microscopic variability. And there are many more, which are summarized in this Nature Review Neuroscience article. So I recommend you look into this review if you're interested in the mechanisms. This is a full pathway from looking a ball approaching to you and actuating your muscles to try to catch it. There are variabilities in doing that particular task. And this diagram basically shows you there's a membrane issue, there's the cellular noise, there's ion channel, there's electrical issues, there are synaptic issues. All of these places have variability. But that's not the only source of variability. Because we're doing an empirical study, there are other variabilities that are epistemological, not biophysical. These are uncontrolled external variables. For example, you don't know where the moon was. Maybe the location of the moon matters a lot to your trial. Joking, but there are certain variables, right? You don't know if your lab mate today played with a cat before coming to the lab and had some molecules from the cat that was different from the other days, right? There are external variables that you cannot completely control. And also, even within the animal, there are many unobserved internal state variables, right? You don't know all the states of all the synapses. From trial 1 to trial 2, maybe some of the synapses have changed their states. Maybe there are more vesicles ready to be released for the next trial because of some biophysical reason. If you knew it, if you have observed it exactly what the synapses were doing, maybe you could have explained the variability with those variables. There are also different levels of neuromodulators that could be possible. From trial to trial, it could be fluctuating. And of course, there are spontaneous activity all across the brain. Maybe the animal was, daydreaming about a carrot or whatever, right? There are spontaneous activities that not the particular neurons that you're recording from are being influenced by those activities. So always keep this in mind when you are looking at these kind of variabilities. Okay, so let's dive into Who Wants to Be a Billionaire. So this is going to be a too short set of questions. Let's say you had a spike train with 3 spikes. You're recording from one neuron. It had 3 spikes. The 2 in the beginning, 1 slightly later. This was in response to seeing an elephant. You showed an elephant, they responded with 3 spikes. The question is, which of the following, A, B, C, or D, were response to the elephant next time? So I showed the elephant exactly controlled. I'm trying to do 2 trials of looking at elephants. So which one of these trials do you think corresponds to the same elephant response? Okay, I'll give you a couple seconds. You can pause the video if you want to think about it a little more. Okay, there was no answer to this, fixed answer to this question. So I'm going to give you a hint. The hint is I had more trials. So this was trial 1. I had trial 2, 3, 4, 5 that I did not show you. These trials were also elephant trials. I'm showing you an elephant. Sometimes it's spiked 2 spikes, sometimes it's spiked 3 spikes, and so on. Now, if I ask you, you would say, oh, it's probably D, because D has a similar property as all of these other ones, right? There's clearly trial-to-trial variability, but the variability is really in missing spikes. There are always spikes that are exactly timed at the same time, 5 milliseconds, 10 milliseconds, whatever, exact precise time, but sometimes they were missing. That's the only variability, but they were all representing, elephants with these temporal response. So we'll come back to this, but this is the temporal code way of looking at things. Okay, I'm going to give you a second case. Same response, elephant response, same choices, but this time I'm giving you these hint trials that were like this. So I'm going to give you 2 seconds, or you can pause the video. Oh, the animation already came in. Yeah, so you can see that the variability structure is different, right? Sometimes there are more spikes, sometimes there are less spikes. Timing is not exactly the same anymore, but there are approximately higher firing rates. There are more spikes in the beginning, no spikes in the middle, and then there's, less spikes at this, later part here. So maybe this corresponds to A or C, perhaps, which, well, C is probably more likely. It has, it looks like it has more correct number of spikes here. So this tells you that the neural code really depends on what the variability structure is, right? If there's noise in certain directions, you should really know what that noise direction is and what the signal direction is. This brings us to the principle of no universal code, the neural tower of Babel. So what is considered similar responses, as we just saw from this Who Wants to Be a Billionaire, differs across neural systems. It differs between different brain areas, different animals, different cell types, different circuits, different contexts. So basically, even if you completely understood what the neural code is for a single neuron, that will not tell you much about the neural code in other neurons or in different brain areas. And the exact same spiking pattern can mean completely different thing for different neurons. Yeah. So that's the basic idea here, and this means that we have to always, rid of our assumptions of what the neural code is, and always pay attention to the data before we make any conclusions. Yeah. So this means both neurons and neuroscientists, right, must be statisticians, because there's noise in it, there's some signal in it, signal itself has some statistical structure in it, and since there's no universal way of reading this out or interpreting that information, they need to learn from all the spikes they're getting or whatever signals they're getting. So neurons are the same as neuroscientists. Neurons have the advantage of having more data, because they are exposed to more information. They're also genetically encoded to do things more stereotypically. So neurons have a slight advantage, but that still means they need to somehow take care of these various representations that might result in different meanings or different information. But fortunately, it's not all completely different. There are some common features, and we will talk about those. So the first common feature is that we rely on very often is the rate code. So the principle of rate code says the firing rate, the average number of spikes, is typically modulated if you have different conditions. If I show an elephant versus I show a cat, it'll modulate the number of spikes. And if you average these spikes, you are removing some of the trial-to-trial variability. The timing doesn't have to be exactly the same. The number of spikes doesn't have to be exactly the same. The average number of spikes is what matters, right? When you average things, things tend to be more stable statistically. And this justifies the way we analyze data. We typically average across multiple trials. Like condition A, we have 10 trials. You average those 10 trials, build the firing rate over time, or also known as the peristimulus time histogram, or PSTH. And so let's look at some examples. So here's area MT again, the middle temporal area that likes visual motion. And these are 15 different trials with the same stimulus. They have some firing rate, as you can hear. And let's look at a different condition. Now this is a trial type B, where the stimulus was different kinds of signal. And you can visually see that they are different. And you can also hear the difference, right? The average firing rate in trial A condition was 24, and average firing rate for condition B was 64, much higher. It's firing rate. And if you systematically change this one variable of how strong the visual motion is across multiple trials, you do a different number of trials. You perform same number of trials for different conditions repeatedly, collect all the different trials for the same stimulus condition and measure their firing rate, what you see on the right is the raster plot for all conditions. You can plot the average firing rate conditioned on that strength of motion. This x-axis here is the strength of motion. If it's a strong motion to the right, it's over here. If it's a strong motion to the left, it's over here. If the motion is very weak or ambiguous, it's around 0. On the y-axis, you have the firing rate. So this particular neuron fires high firing rate, 64 spikes per second, whenever this motion is to the right. So there's a tendency, there's a curve here that shows you the encoding of this information. And this is called this is an example of a tuning curve. A tuning curve is a very important concept and object that you would encounter many times. And this particular tuning curve here is a tuning curve of this empty neuron's firing rate respect to this strength of motion, right? So this is a function. It's a firing rate function as a function of this parameter, which is the strength of visual motion in this case. So I'm going to give you another example of an empty neuron. This is a different neuron with slightly cleaner tuning curve here. You can see it's less noisy and it still likes, strong rightward motion in this case. And tuning curves are not just for visual motion. Now, in this example, I'm showing you a tuning curve for orientation. So in primary visual cortex, V1, if you look at a single neuron, they really like grading. So whenever you have a bar or a set of bars, like a grading, they tend to fire depending on the orientation of the bar. And in this neuron, the firing rate tends to be high. It likes to fire whenever you show a orientation of this blue kind, and it fires the opposite orientation, the 90 degrees orientation, this orthogonal orientation, the red guy here. This neuron does not fire very much. So this curve here, again, is a tuning curve, and it depends. If you know the concept of receptive field, this is a function of a receptive field. Tuning curve can also be an auditory cortex or an auditory nerve. In this case, this is the output of the cochlea. This is what encoding sound directly from the cochlea, sending it to the downstream areas, the auditory thalamus, and then to the auditory cortex. And these neurons, in this case, a very simple tuning curve here, where the sound pressure level, the SPL, if it's high, this auditory nerve fires more. So the firing rate on the y-axis again. So this curve shows you at some point of loudness, this neuron certainly fires more and more spikes, and then it saturates at some point, right? So this is a tuning curve. You can plot the empty tuning curve in a circular manner as well. This is called a Rose plot, just to make sure that you are familiar with such a plot. This is a stimulus direction. So empty neurons do like visual motion, as we discussed, but they like the visual motion in different directions, right? Previously, the tuning curves I showed you for empty neurons were as a function of motion strength. How strong is the motion? How robust is the visual motion on the screen? Now here, the motions are strong, but they are in different directions. So the motion of this random dots moving upwards versus left or to the diagonal different directions are represented around the circle. And this solid line in the middle shows you the firing rate. So if it's close to the center, it means the firing rate is 0. If it's further away, that means the firing rate is high. So this empty neuron particularly likes these directions that are up to the up direction or slightly to the right, and it does not fire very much on these other directions, the opposite directions. This is an example of rostral superior temporal sulcus. In this area, it's a ventral stream of visual processing areas where you have these neurons that like different viewing angle of a head. So what you're showing here the x-axis is different angles of a monkey head, the picture of a monkey that you're showing to the animal, and the monkey, this particular neuron responds very well whenever the monkey is looking to the right. But well, the monkey that's the STS neuron that's being recorded is from the monkey. This monkey is looking at a picture on the screen. If the picture of the monkey is looking to the right, then this neuron fires a lot. And this bottom neuron has these two bumps. It likes either left-looking or right-looking pictures, but not back of a monkey picture or front of a monkey picture, just both directions. So these are different ways of building these kind of encoding models. You're analyzing your data, trying to represent it as, oh, this is the neural code, potentially a neural code, because it modulates by the variable that I'm changing. I'm controlling this variable, the firing rate changes. So this is a rate code, and the firing rate changes in such a way that it's modulated in a systematic fashion. I'm writing that down as a tuning curve. Okay, so that's tuning curve. Let's revisit noise a little bit and how to do some basic statistics. So if you add some noise to a signal, what happens? So let's listen to some audio, clean audio, and some audio with some noise.
Neural computation is awesome.
That was clean.
Neural computation is awesome.
I'm adding 0.01 variance noise to the signal. Now I'm going to add variance 1 noise.
Neural computation is awesome.
So this ratio between the signal power and the noise variance, the power of the noise, is called the signal-to-noise ratio. And as you can see, as a signal-to-noise ratio gets worse, your noise is stronger than the signal, then it's very hard to tell what's happening here. But if you have this kind of noise, variance, well, here's, in case you forgot what variance means, this is the estimator for variance. Variance is a deviation from the mean, but measured in terms of its squared. Right. This is what variance is. So if you have noise that does this, then you can always average things, assuming your noise is really noise. It's very independent of your signal. And you can try to remove noise or reduce the amount of noise. So that's the idea of averaging. So let's see a simple, let's observe this simple mathematical fact here. So if you average across trials and you assume the noise is zero mean, additive, and they're independent, while your signal is deterministic and time-locked. So every time you do the trial, the signal is exactly the same. Every time you do the trial, noise is random every trial, right? So that's what we're doing in an experiment, and we're going to average across trials. Right? We're taking we're computing PSTH, for example. So the signal is, for every trial, the signal is S. It's the same for every trial, but the noise Ni is different for every trial. That's why you have a subscript I. We're summing up all of these trials, n of these trials with the summation here, dividing by n. Signal is summed n times and divided by n, so the output is just signal plus noise term, which are summed up and then divided by n. So the question is, what is the variance of the noise, right, after this averaging process? So if you just look at the noise term, the variance of this noise term, because of the property of the variance function, it takes 1 over n square out, and then there's a summation, which gives you a factor of n. So the variance itself, if after this process, decreases by a factor of n. So if you have 100 trials, the noise is reduced, the variance of the noise is reduced by 100 times, or equivalently, the standard deviation of the noise is decreased by 10 times, right? So this is a good way of removing noise if you have very noisy signal, such as noisy spike trains. But of course, the brain can't use averaging across trials. That makes no sense. The brain has to function, the neural system has to function every trial. So it needs to function on single trials without averaging across trials, somehow doing something different. So how does it do it? So keep that in mind while we observe a property of the noise in neural system. So one thing is that this spike count variance. So if we're measuring the spike count and looking at the variance, it actually is not additive as we just assumed. So in this analysis, what we were assuming was this additive noise is always the same. It does not depend on what the signal was. But in fact, if you look at the real spike count, in this case of fly and cat, depending on the neuron, depending on the condition, these are different spike counts. And if you look at the spike count across many trials, repeated trials, and so these are, firing rate 3. There are 3 spikes on average for this particular condition and neuron. And you look at the variance of that neuron, it's around 1 or 2 or something like that, right? So if you collect all of these different conditions and neurons from these flies and cats, they tend to lie on this line, which is not what we typically see in like audio transmission, for example, or telegrams, where signals are typically additive. This looks more multiplicative, right? So instead of the noise variance being just constant over different conditions, it depends on the mean. So really, we shouldn't just talk about the variance on its own, but we should talk about this ratio between the mean and the variance. And that's the concept of the Fano factor. So this pops up whenever you are trying to quantify, is this neuron reliable or not, right? And one thing you want to compute if you have multiple trials is the Fano factor. It captures the trial variability with a single number. In this case, I'm writing it as FF. It's a ratio between the variance of the spike count across trials divided by the mean spike count across trials. So the denominator is the mean firing rate, and then the top here is the variance of the spike count. And as I just showed you earlier in the previous picture, it was a paper, a science paper. This Fano factor tends to be around 1-ish. It depends on the neuron, it depends on the animal, it depends on the task, of course, but it's around 1-ish. Some neurons are much lower than 1. But and this Fano factor equals 1 is actually the condition for Poisson process as well. So if you have seen Poisson process used as a surrogate for spiking noise, this is one of the justifications. Anyways, so if Fano factor is close to 0, that means the variance was very small. It scales with mean, still scales with mean, but the variance was very small. So it's a reliable neuron. If the Fano factor is large, if the Fano factor is 10, your signal is always going to be buried in your noise, right? Okay. So let's hold on to, let's stop talking about the rate code for a moment and visit a different kind of code, a temporal code. Temporal code is defined as the opposite of rate code. So just to reemphasize what a rate code was, a rate code is a hypothesis that says all information is represented in the firing rate of neurons. It's the spike count, average spike count that matters, and that contains all the information. The precise timing patterns, the small deviation in spike counts, they don't matter too much. It's the firing rate. If the firing rate is 40 hertz versus 20 hertz, it means A and B, right? That's the rate code. It's the extreme case of extreme interpretation of rate code. And temporal code is basically saying, no, you're too strict. There must be some information that's not in the rate on its own. So typically people will say temporal code, they mean precise timing of the spikes matter. So if I have 2 spikes that are exactly 5 milliseconds apart, that's different than 2 spikes that are 7 milliseconds apart, right? That's one kind of temporal code. temporal code makes sense mainly because neurons can be quite precise. I mean, we talked about different reasons why neurons cannot be precise in their response, but surprisingly, even in cortex, here this is a single auditory cortex neuron in a rat in response to a tone. So this is 28 kilohertz tone. It's a beep sound. You can't hear it. It's too high of a frequency. But whenever that tone shows up, this neuron fires exactly one spike every single trial. It's a very precise machine here. Yeah, the auditory system is especially more precise like that. And this neuron in the bottom, slightly less reliable because it's missing a spike sometimes, but it fires exactly once or none in response to these tones. Yeah, so here, quick puzzle or quiz. What is the Fano factor of the trials in this condition B, the top one? Let's say we took a window, this whole window. What's the Fano factor? Pause the video if you want to think about it more. Yeah, although there's a little bit of jitter, if I take the full window to compute this, to count the number of spikes, the number of spikes is always 1. So the average number of spikes across trial is 1. And the variance is the deviation from the mean, right? How much is each trial deviated from that 1? Trial number 1 has 1 spike. The mean was 1, 1 minus 1 is 0, so its deviation is 0. Square of 0 is 0. And averaging that across trials is still 0. So the variance across trials is 0. There's no variability across trials. It was always reliably giving you 1 spike. So the Fano factor is 0. The most precise it could be. The lowest Fano factor you can have. So yeah, so there are neurons that are quite precise. And it's not just auditory cortex. If you look at a pyramidal neuron, and this is layer 5 regular spiking neuron, in this case, yeah, well, this is let me grab some water. It's not just auditory cortex. So if you look at layer 5 regular spiking neuron here, this is a famous study by Meinen and Serginovsky, where they injected current, whole cell patch clamp situation. You inject a current. This is the trace of the current. It's about 1 second long here, and you record this firing of this neuron. And if you look at it, if you repeat the same stimulus, how many times was this? 25 times. The response is almost exactly the same. You have exact same timing of the spikes, exact same membrane potential fluctuations, right? Oops. Exact same membrane potential. And if you look at the exact same timing of spikes and membrane potentials. Not only in layer 5 regular spiking neurons, this is now an example of a lateral geniculate nucleus. This is where output of the retina, the retinal ganglion cells send their spikes here. And if you look at these neurons, these are multiple trials watching a movie that has this, intensity of over time. They also fire very, very reliably. So there's an interesting thing that we can try to do to see experience this directly. If you have headphones, put on your headphones both ears, because this is going to be a sound localization task. I made this up by coding up some MATLAB. Okay, here we go. Your job is to tell me which direction the sound came from.
1, 2, 3, 4, 5, 6, 7.
So note that the volume of your left ear and right ear, the sound is exactly the same. In my MATLAB code, I generate the sound All I did was shift it a little bit in time. Let's hear it one more time.
1, 2, 3, 4, 5, 6, 7.
I think the volume was a little too high. Let me quickly fix that. Okay.
1, 2, 3, 4, 5, 6, 7.
Okay. Yeah, so there's one way we can actually experience this kind of precision in firing, especially in the auditory cortex ourselves, which is a sound localization task. So if you have headphones, put in your headphones. It needs to be both ears. And I'm going to play a set of sounds. Tell me if the source of the sound is moving from left to right or right to left, or is it just stationary, is it up and down, back and forth and whatnot, okay? Playing the sound now.
1, 2, 3, 4, 5, 6, 7.
All right, so the sound what I generated in MATLAB, the volume is not different. It's not like you're hearing one side is louder than the other. That's why you think it's one side or the other. It's just the timing, okay? I'm gonna play it one more time. If I can play it one more time. Oh, here we go.
1, 2, 3, 4, 5, 6, 7.
Okay, hopefully you heard that the sound location, your perceived sound location is changing. So why does this work? It works because there's timing difference between the left and right ear. A signal that's coming to your left and right ear is called the interaural time difference, or ITD. And if sound is coming from the front, if the source of the sound is from the front, it'll reach both ears at the same time, right? Because there's symmetry. But if sound is coming from the left, there's a sound travels at finite speed, and your head has, head is not flat like a paper. There's some distance to travel from left to right. So that distance that it travels is around a standard human head size, 640 microseconds. And this is the extreme case. You can tell the difference if the source is slightly off. If it's 20 degrees off, it's 200 microseconds. And yes, I did say microseconds. So the actual potential itself it has a width in the order of a millisecond, 1,000 microseconds. So this is very small difference in the timing of sound, which has to be encoded as spikes in the auditory nerve, sent to the brain, in this case, brainstem, and has to do some fantastic, reliable computation with those spikes. So yeah, so temporal code makes some sense in some situations. Not always, but yeah. You need to keep in mind, you need to keep your heart open in case your data has typical patterns of temporal code that you don't want to lose because all you did was to average across trials because you thought rate code was the king. All right, let's switch gears and get to population code. We talked about mostly single neuron at a time. Now let's look at a code that's distributed across many neurons. And for this, we're going to go to the motor system. Okay, so here is a diagram of a single neuron. I'm going to show you a single neuron first, of a single neuron in the motor cortex of a monkey in relation to a center out reaching task. So the monkey is holding a joystick and it's moving its arm in different directions. So the target comes up and it needs to move that joystick to a particular direction to get a reward. And whenever they make that motion, this is the timing of the vertical line in the center is the timing of their motion. There's spikes in this neuron that fires, and you can see that in some direction there are more spikes, the other direction there are less spikes. You can plot it as a tuning curve on the right hand side. The firing rate for reaching toward a 150-degree direction has higher firing rate and whatnot. So this is nothing surprising here. But if you try to, so this is the encoding model. But if you try to use this to decode, right? So let's say for a single trial, you got 30 spikes from this neuron, and you look at this diagram and you say, oh, 30 spikes, it must be, oh no, it could be either 65 degrees, or it could be reaching for 250 degrees, right? It's ambiguous what the original movement is using just one single neuron. But if you have many neurons with many different tunings, this ambiguity will go away, because the unique combination that's possible is not going to overlap. There's always going to be a unique firing pattern for every particular direction. So here's an example of a decoding system where the monkey's motor cortex, so there's an electrode area in the monkey's motor cortex, spikes coming out, there's a decoder that tries to decode this signal and translates that to this robotic arm that has 4 degrees of freedom. There's 3 in the arm, and then it can do this grasping thing. It's trying to grab this marshmallow in 3D and bring it to their head. You can kind of see the cable dangling there. That was the recording electrode cable that goes to the amplifier. And this is a very early study, and a lot has happened since then in this particular area of decoding motor cortex, but the principles have not changed too much. The basic principle is to look at define each neuron having a single preferred direction, right? So if you have a tuning curve, you would say, oh, this neuron likes this direction. It fires the most whenever the direction is towards the right lower corner. So you would tag that neuron as, oh, this neuron likes that corner with that single preferred direction. You'll notate that with a single preferred direction. And when you have a lot of neurons with different preferred direction, each neuron having different preferred direction, what you want to do is do this population decoding method, population vector decoding method, pioneered by Georgopoulos. And what you do is you are weighting each vector that you get from each neuron by analyzing their tuning curve. And then in a single trial, when you get firing rate, neuron 1 fire 10 spikes, neuron 2 fire 20 spikes, you're going to weight each vector with that corresponding firing rate, and then just average all the weighted vectors together, which is called a population vector. It's a very simplistic method, but it works pretty well. So for example, this is just a cartoon example. Let's say you had 8 different directions. Each neuron has different preferred direction 1, 2, 3, 4, and so on. And if neuron 2 and 3 fired a lot, while all the other neurons didn't fire very much, the weighted vectors would have this kind of shape. And then you will average all the vectors together to get the population vector red one. And then you would send that command in that particular video, command to the robotic arm that says, oh, arm, move that direction, or change this joint, change that joint, change your opening of the hand. Each of them with a different population vector decoder. And it works pretty well. Even the modern ones are extensions of this method, more sophisticated algorithms analyzing this, but the fundamental idea is pretty similar. Okay, let's switch gears and get to the next principle, the principle of noise correlations. So here, what we're seeing is 2 neurons. So before, when we talked about noise and trial-to-trial variability, we were talking about noise in a single neuron. Now we're going to talk about noise that are correlated across multiple neurons. So this is neuron 1's firing rate on the x-axis, and the y-axis is neuron 2's firing rate. So each dot here corresponds to a trial. So there are many trials here, and there are two different trial types. Trial type A and B. One is red colored, one is blue colored. And as you can see, whenever there's a blue trial, there's a variability. There's variability in neuron 1's firing rate. There's variability in neuron 2's firing rate. But the variabilities themselves are correlated such that it's elongated in this positive direction. The same for this red one. Right, so we call this a positive noise correlation. That's the lingo we use in neural code. And this ellipse here, it just represents that noise correlation as a variance, as a covariance. Okay, so let's look at these two examples. These are simulated neurons here. That's why we can have such a clear data. In the left case, we have positive correlation. On the right case, the average is the same. Mean firing rate for condition A, this diamond here, and mean firing rate for condition B, this blue diamond here, are the same from left and right conditions, but the noise correlation is different. Left one, the noise correlation is correlated. The noises are positively correlated, and the right, the magnitude of noise per each direction is the same, but the noise is not correlated. And as you can see on the right-hand side, there are some trials that become more ambiguous, right? In this region that are overlapping, you can tell if it's a red trial or a blue trial. It's hard to decode if you try to decode just from these just two neurons. In contrast, on the left side, this positive noise correlation somehow is helping you, and the blue dots and the red dots are more separated. If you build a decoder, you can decode better in the left case than the right case. So positive noise correlation is helping. Okay, and if you have negative noise correlation, if you have same signal, so left diagram is the same, the right diagram, the signal is the same, the mean firing rate for neuron 1 and 2 for each condition are the same, but now the noise correlation is negative correlation. Right? Whenever there's a variation from the mean from one trial to the left, the noise on the neuron 2, so whenever neuron 1 fires less than the average, neuron 2 tends to fire more than the average. Their sign is opposite. That means this is a negative correlation. Right? You see it's a diagonal that's has a slope of negative slope. So this negative correlation hurts you. You can't really decode. The overlap is even more. So positive correlation is good, negative correlation is bad. Is it always the case? And the answer is no. If you want to think about it, pause the video. But the thing is, it depends. It depends. So in this particular example here, it looks similar. The situation looks similar, but as you can see, negative noise correlation, which is a middle diagram here, helps you decode, while positive correlation makes you more confused. It's harder to decode. So why is this the case? It's because this positive correlation itself is in the direction of the difference of the mean, right? The blue cloud, the condition B versus condition A, these red cloud versus blue cloud is in the direction, it's positive correlation direction. And the noise itself is in the same direction. So this is a bad noise, sometimes called a information limiting noise, because the noise itself, if you have a certain noise, it makes you look like you're changing from condition A to condition B. And that really depends on what is the difference between condition A and condition B. So whichever condition A and condition B exist in the space of tuning curves or mean firing rates or what have you in the neural code space, that direction, if you have noise in that direction, that's a bad noise because it mimics the signal. You have a noise that looks like a signal, you can't get rid of the noise. Okay, so that's the moral of the story. Okay, switching gears once again. Now we talked about decoding a little bit. Decoding, a lot of people love decoding, but in the field, unfortunately, there's a tendency to over-interpret decoding results. You're measuring from a particular brain area, you decode how wakeful they are, and you're like, oh, I found the brain area that encodes or that represents how awake they are. It may or may not be true. Let me give you an example of how this does not work in an extreme case. So let's say you had dog and cat images encoded by the eye, the retina, and then you have a population spikes coming out of the retina. And if you're smart enough, you can build a powerful decoder. Let's use machine learning, deep neural network to take all of these spike counts across the retina and then decode, dog or cat. We can do this. We know we can do this pretty easily because we can do it in the pixel space. Why not in the retinal ganglion cell space, which is not, it's just a filtered version of it, more or less. So this is possible, but this does not mean the retina knows what dog and cats are. Retina doesn't know this. I mean, if you train an animal to do this task, they will be able to do it behaviorally, but not thanks to retina being trained. It's the ventral pathway and the visual system, many downstream layers down the road. They might be able to have this decoding capability, but not at the retina, right? So, but if you claim that because I can decode if it's a cat or dog at the output of the retina, I know how dog and cat information is encoded in the retina, that's a wrong conclusion. So be careful. This is a very common mistake people make. So you need to be very careful. It's an evidence towards it, but it's not a conclusive thing at all. Another thing is both encoding and decoding analysis are really just capturing structure in the data. You had two variables. You had some neural responses, your neural signals, and you had some variables that were information of sorts, and they were dependent, right? One changes, the other one changed. There's some relationship. But that doesn't mean they're causal at all. So a particular example here is, let's say you have a variable that's encoding neural activity. It might just be correlated with what's actually causal. So the example here is, let's say you have some signal coming in. Let's say call that some signal coming X coming in. Neuron 1 does something with signal, produces behavior Y. You capture Y. And then you're recording from neuron 2, which is not causal to this computation. The transformation from X to Y and then making the behavior, that relationship was really encoded and computed by neuron 1. But neuron 1 just happens to also send this information to neuron 2 as a copy. So if you happen to put your electrode in neuron 2 and record from neuron 2, and you find that, oh my God, I can decode Y, or I found an encoding scheme. The firing rate changes in neuron 2 whenever I change X. Yes, those are true, but that does not mean neuron 2 is in any causal chain in computing this quantity. Or, yeah, it's not really, I mean, it could be encoding it and using it somewhere else, but if this is the whole circuit, neuron 2 is not very important. Right? So you need to be careful. All right, this is the last principle that I will go into the detail. This is the principle of efficient coding, proposed by Horace Barlow. Cut. Sorry about that. Okay, so this is the last principle I will discuss in detail, which is the principle of efficient coding, proposed by Horace Barlow in the late 50s. He made various remarks. For example, he made this remark, which is interesting. A wing would be most mystifying structure if one did not know that birds flew. In analogy, if you not know, a neuron's firing pattern would be most mystifying if you don't know what their purpose was. What is their goal? What's the overarching objective of the system. Why is the neuron firing the way that it's firing? Why is it encoding signals in certain way? So these kind of theories of how the brain works are called a normative theory because it's top down. And what Barlow said was the overall this overarching objective of neurons in early sensory system, for example, is to encode as much information as possible and relay that to the later areas. And he quantified this in a particular equation. This is called the redundancy reduction or the efficient coding hypothesis, where you have a channel. Let's be more concrete. Let's take the optic nerve. You have the retina output, which is the retinal ganglion cell spiking pattern that is sent to the visual areas through the optic nerve. And the optic nerve has a finite capacity. There are way more neurons in the retina than there are neurons in the optic nerve. So there must be something lost, right? So there's a channel capacity, C here. And what we're trying to see is, let's try to maximize the utilization of this channel. Let's not send any redundant information. If I send the same information twice, that doesn't make any sense. We need to try to make sure that all the information we send through the optic nerve is distinct, right? As as high of an entropy as possible, because this whole framework is based on Shannon's information theory. We can capture this randomness or information, amount of information with entropy, this quantity h of y here. So assuming there's no noise, the objective of optic nerve, whatever is outputting things to the optic nerve, should try to maximize their entropy, is what the conclusion is from this. And more modern version of this can be understood by looking at oh, wait a second. Yeah, through the optic nerve. Let's look at an example. This what does that mean? Its implication that we'll not go into the details of the math or information theory here, but looking at the implications are kind of interesting. So let's look at natural images. Natural images are highly correlated in space because all objects are big enough to have smooth surfaces. Pixels that are nearby typically has the same value. If you see a red pixel, next to it is typically a red pixel. Small variations, but it's unlikely that a black pixel is next to a red pixel. So that could be captured by this diagram here, where pixel I versus pixel I plus 1, these are two neighboring pixels. If you look at multiple images, these pixels would tend to have a high correlation and small amount of variations around it. And what efficient coding says is, hey, if you're encoding pixel I and pixel I plus 1, if you send both information, you're sending duplicate information. Try to find, if you're sending pixel I, when you're sending pixel I plus 1, don't encode pixel I plus 1. Try to decorrelate it as much as possible and send more useful information, perhaps in the direction that's not in pixel 1's direction, right? So what you want to send is a distribution like this that has maximum entropy. This means the output of the retinal ganglion cell, for example, would have to use its full dynamic range, and then it has to decorrelate wherever it sees correlation, because correlation means redundancy, and redundancy is bad. More modern versions of this can include noise. So if you have no noise, you want to decorrelate at maximum, but if there's some noise, you don't want to send noise. The thing is, what is important to send is signal, and as much as you want to not send duplicate signal, you don't want to send noise instead of signal, right? So whatever you can do to suppress noise. For example, if you have in a higher noise case, you want to average across space a little bit, smooth it a little bit such that noise goes away, and then send that information instead. So these principles explain a lot in terms of encoding in early sensory systems, and have had a huge amount of influence in understanding decorrelation, information maximizing codes, sparse coding, predictive coding, adaptive codes, independent component analysis, and Bayesian efficient coding, which is my own. Also, it influenced a lot in machine learning, where it's used as a particular kind of objective, information theoretic objective function for training different models. Okay, so let us summarize all the principles and with additional bonus principles. Let's just quickly review. Neural signals are variable, huge amount of trial-to-trial variability, and there's no universal neural code, which means both the neural system and neuroscientists have to be somewhat of a statistician to understand what's going on and operate in that realm. An important aspect is that firing rate carries some information, if not all. If you say it's all, it's rate code. If it could be mixed with some temporal code. But this is very important that firing rate should be looked at first, because there's always some information there and easy to analyze. So tuning curves and PSTHs are great to look at. We talked about how if you have more than one neuron, information can be spread across neurons. In that case, you have a population code. There are multiple ways of decoding and encoding in population code. We saw a simple case of population vector, and we also discussed different structures of noise in the population can be good or bad. Noise correlations that are in the direction of signal are limiting. So these are the information limiting bad noise. There were a couple of caveats. Decoding does not imply causality nor neural availability. And if you really want to know the causal relations, decoding, encoding analysis is not going to cut it. You need to be able to do manipulations. We didn't go into the details here, but if you perturb the system or do some manipulative experiments, you could improve upon your causality, and sometimes anatomical experiments and knowledge would also help you with that. We talked about efficient codes. Efficient codes reduce redundancy. It makes everything more efficient in a sense, and hence you don't have to use a very big channel to send information. We didn't talk about these last 2 points, but just quickly, in terms of neural availability, we know that decoding does not always give you neural availability, but a good proxy of neural availability in a small brain area is a linear code. So if you do, if you do a linear regression or some simple linear decoding, typically that's not too bad, right? But if you fit a deep neural network, something very complex as a decoder, then that can be very, very misleading. And the last point here, which is important, is that if the system learns, because we're only talking about short-term, right? Neural code is about short-term representations. If the system learns, the neural code changes. And that can have a very interesting cascading effect that complicates the matter a lot. All right, so that's it for the lecture of Principles of Neural Coding. I hope you learned something. And if you want to read further, here are some reading materials for you. Bye.